My name is Jonathan Kaufmann. This is our paper, One Class Ensembles for Rare Genomic Sequences Identification. This paper was written by me, as well as Kathryn, Asalone, Roberto, Corizzo, Colin, Saldanha, Bracht, and Nathalie Japkowicz, and was a collaboration between the American University Computer Science and Biology departments. We were all at American University at the time it was written. As an overview for what we'll be talking about today, we'll be talking to you about the problem that we faced, the background between biology and machine learning, the methods including our data and models, the results from several experiments we ran, and our conclusions and areas for further research. The problem we faced was identifying something known as the germline-restricted chromosome, or GRC. The GRC is different than what most people think of with chromosomes, because in biology class you were taught that every chromosome in every cell is the same. Right? DNA is about the same, you have the same number of chromosomes everywhere. But this isn't true in all animals. In specific species of songbird, they have an extra chromosome in their germline or reproductive cells, versus the somatic cells, which are just the normal cells from the liver, the heart, the brain, etc. These cells all have the same chromosomes. The germline cell has all of the normal chromosomes plus one. This chromosome has never been sequenced. In particular, we looked at the zebrafinch in this research, and the genome project involving the zebrafinch only sequenced somatic cells and no germline cell. In terms of the biological background, only 2 genes from the GRC have been identified, which is a very, very small number. The germline restricted chromosome can't be isolated using conventional methods due to its size. It's the largest chromosome in the zebrafinch, and it makes about 10% of the total DNA sequence complexity in the bird. We don't really know what the purpose of the GRC is. It's thought maybe that it's involved in some ancient processes that aren't really relevant anymore. We do know that it's passed exclusively maternally and not at all paternally. From a machine learning side, in this paper we use one-class classifiers, which basically means that these machine learning models are trained on only one class of data, either negative or positive, versus trained on both, as in supervised learning. And the idea is that we can train it on one class of data so that it can identify observations belonging to the other class, that is, not belonging to the class it was trained on. And the paper today, this particular problem is a canonical case where this makes sense, because we have the somatic cells with all of the normal chromosomes, and then we have a mixed data set in the germline with both all of the normal chromosomes and the germline restricted chromosome. The idea being that we would be able to separate the germline cell or the germline chromosome from the somatic chromosomes that are present in both the normal and mixed cell. So in general, one-class classifiers don't perform as well as supervised learning. They obviously have less information, but oftentimes when you use them, they're the only option, as in this case where we have very, very, very few true positives. There are many one-class models, including one-class support vector machines, autoencoders, local outlier factor, and more recently, isolation forest. In terms of our data, we took DNA from a zebrafinch. As I explained before, the true negative data set is the somatic or liver cell. This was data taken from a liver in this case, and there were tens of millions of sequences. Our evaluative data set was a germline or reproductive cell. In this case, it was a male zebrafinch, which again has tens of millions of sequences. And from that, the biologists that we worked with constructed a hypothetical GRC set with about 13,000 sequences. And these were sequences that they believed were likely to belong to the GRC, but they really weren't confident in it. And so we were hoping to determine whether or not they actually were in this paper. We also constructed a small true positive data set based on those 2 sequences I told you about before, identified in Biderman et al. In 2018 and Ido et al. In 2009. In terms of our data preparation, we split the sequences into subsequences of size 60 so that all of the sequences would be of the same length. We also separated, obviously, validation and testing data sets. Validation sets we tune on, the testing data sets we don't, in order to evaluate our models. And in that case, we had 10,000 negative observations in each of those data sets. and roughly 90 positive observations. There were only 180 positive subsequences in total. And our feature that we selected were engrams, which is called often K-mers in the biological literature. In this case, we selected a 4-gram, and since there are 4 letters in the DNA alphabet, there's a feature space of 256, and both the subsequence size as well as the 4 grams were selected based on experimentation, where we found that if we had a lower size, we would lose accuracy and lose performance in that metric. But if we had a higher size, we wouldn't really gain any more in terms of accuracy, but we would lose in terms of computation performance, and it would just become a lot slower to perform the computations. So we used 2 models in this case, both one-class learners. The first is local outlier factor, which bases anomaly scores on local density of an observation and compare it to its neighbors. And the idea here is essentially that if you take an anomalous data point and place it among normal data points, it is going to be less locally dense as opposed to the others. With the isolation forest, you construct a series of trees by cutting the data to isolate points. And the idea here is very similar, that isolating an anomalous data point will be much easier than isolating a normal data point. So we created 5 of each of these models and combined it into an ensemble of a total of 10 models. We trained each model on 10,000 observations, and again, that was based on experimentation. We found less lowered our accuracy, more didn't increase it, but did substantially increase computational time. And then, obviously, because the local outlier factor and isolation forest don't compute anomaly scores in the same range, the isolation forest is bound between negative 0.5 and 0.5. Local outlier factor is not. We had to construct a transformation constant. We actually constructed 4, 2 for each model, 1 for observations that fell below 0, and 1 for observation scores that fell above 0 for each of the model types. And that was constructed by taking a large amount of anomaly scores from normal observations and then normalizing them such that 95% of the ones that fell below 0 were between 0 and negative 0.5, and then 95% of the ones above 0 were between 0 and 0.5. And in order to generate a subsequence anomaly score, we averaged across the anomaly scores generated by the models. We also tried a specification where we dropped the highest and lowest score, but we found that that didn't really change performance at all. And then, of course, the sequence anomaly score was just an average of the subsequent scores. In this case, it's effectively an average of averages. So our first experiment was to compare our model against other specifications. We selected a single isolation forest model as well as a single local outlier factor model, as well as ensembles of a particular type, ensembles of 10 local outlier factors and 10 isolation forest models, and our performance metric was the area under the curve, or AUC. We see in the validation data set, the one we tuned against, that the ensemble models, as expected, perform better than their single counterparts, and this is, we assume, because they're better able to reduce the errors that a single model might make by having a wider breadth of experience in terms of the different models. And the mixed ensemble outperformed both of the ensembles of a particular model type, and again, we suspect that that's because the different model types have different errors that they're prone to make, different biases, and this helps correct for that. In our test data set, we see exactly the same thing, and that our model performed better than all of the others. The second test that we decided to conduct was a comparison test. So we generated anomaly scores for all the data sets, the hypothetical GRC, as well as 10,000 negatives and all of the known positives, all 183. And first we wanted to compare the known positives, the known GRC, versus the known negatives, the somatic cells. And the model should be very clear about which belongs to which. It should be very obvious, looking at a graph of these scores, that the model is able to differentiate these. And secondly, we wanted to compare the hypothetical GRC sequences and that anomaly score distribution against the negative distribution and the positive distribution. And ideally, we would see that the hypothetical GRC, if they were all actually part of the GRC, would match the positive distribution and be differentiated from the negative. So we see in that first bit that there's a very clear difference between the distributions in the negative sequences shown in the blue here and the positive sequences shown in the orange. Now obviously the orange, the positive sequences are relatively sparse, so the graph isn't as filled in, but it's very clear that there's a single peak here, that it's in a nice shape. We see something somewhat similar in the liver distribution, in the negative distribution as well. In the hypothetical distribution compared to the negative in the liver, we see that, again, it's very clearly differentiated, but in the hypothetical GRC, we see a bit of a bimodal distribution where we have a large number of observations that are more anomalous. Again, a lower anomaly score means more anomalous than the negative observations, but we also see a second peak sort of at a higher area there that looks almost very similar to the negative distribution, and this suggests to us perhaps that there are some negative observations within the hypothetical GRC, that not all of the hypothetical GRC sequences actually do belong to the GRC. And lastly, looking at the known GRC, the true positives in orange there against the hypothetical GRC in blue, it's very obvious to us that the distributions look fairly similar, and this is definitely confidence-inspiring. Lastly, we performed a blind test. The biologists, in this case, found a link between one of the sequences in the hypothetical GRC and one of the known sequences in terms of its genetic expression. And so they decided to experimentally validate that gene. And based on their results, which you can see here, it's very clear that this is present in the GRC, or in the germline cell, but it does not appear to be present in any of the somatic cells. And as a result, it's pretty conclusive to say that this is a GRC sequence. So this particular sequence had an anomaly score of 0.03, so just a little bit less, or a little bit lower of a score than the first peak in the hypothetical GRC. It was about the 27th percentile in terms of the anomaly scores, which means that there are about 3,700 observations that appear to the model to be more anomalous than this one. And that suggests to us that there are thousands of potential GRC sequences in that hypothetical GRC dataset. So in conclusion, we believe we've identified thousands of potential GRC sequences, and this research clearly demonstrates the value of machine learning in the biological domain. It shows that researchers can use these tools to focus on the most likely positives when their resources are limited, and how that can help them. And I think it opens up more doors in terms of using these sorts of tools in the future to aid researchers in the space. As far as avenues for future research, we'd like to experimentally test a larger number of sequences that allow us to determine an anomaly score that would be able to differentiate between a germline and restricted chromosome sequence and a non-germline restricted chromosome sequence, a somatic sequence, with a high confidence, say 95 or 99%, and classify all of the germline data such that we could differentiate tens of thousands or maybe even hundreds of thousands of additional GRC sequences that weren't previously known. And this research could also be applied to other similar chromosomes. For example, the zebra finch has another maternally passed chromosome, the W chromosome. That would be another good candidate for this. That's the end of this talk. Thank you for listening.